Hey there, you guys. As promised, I wanted to do a little bit better job lecturing over the last 2 pages of notes in your interactive notebook. That would be right-hand pages 46 and 48. So this is where we would have started off on page 46. Previously, I had gone through conditions, abnormal conditions of the heart, like having an enlarged heart, looking at varicose veins, aneurysms. So this is picking up right after that. So I believe it's page 46, patterns of circulation. We've talked a lot about systemic circulation and pulmonary circulation. So when we talked about our vessels in our previous lecture, sorry, I'm clicking around so much, systemic circulation starts off at, if we start with the left ventricle, then we go to the aorta, getting to smaller arteries till we get to finally the smallest arterioles. And at the capillary bed is where we have exchange happening. And that's the only area where we have exchange. So gas exchange, carbon dioxide for oxygen, and also nutrient exchange for waste. Blood then returns back to the heart through venules, getting larger veins to eventually the vena cava and then into the right atrium. From the right atrium, then we would go to the lungs, right? And then back to the left atrium and ventricle. Sorry, yawning. Going back to the heart. So that's pulmonary circulation. This is systemic, right? And we looked at our blood vessels and how those transfer blood around. So pulmonary circulation, this is from the heart to the lungs and back to the heart again. We've already talked about this quite a bit. When you did your heart dissection, this was the vessel, the pulmonary artery, where you were supposed to cut in to get into the right side of your heart. And a lot of you had a hard time finding that, didn't you? So make sure you go back and review those heart structures again. So pulmonary circulation, I feel like we have covered that pretty well. Hepatic portal circulation. I glossed over this in a couple of the periods really quickly. And I think that I want to just revisit it again here really quickly. Maybe it made perfect sense to you in reading the textbook, maybe not. I've got down here on the bottom a video here that you should watch. It's Armando Hasudigan. He's the instructor that talks and draws in fast time. And his drawings, I wish I could draw like he does. So hepatic portal, hepatic means pertaining to the liver. And this is a unique type of circulation whereby blood drains from the GI tract, which is going to be the colon, and parts of the intestines. Why am I yawning so much? Sorry, you guys. And instead of that blood draining directly back into it, so it goes into major veins from your gastrointestinal tract, and instead of it just making its way back to the vena cava and heading right back up towards, back into the right side of the heart, some of the lung goes in through this portal vein, and it goes into the liver. It's kind of hard to see here with the purple. Okay, but this goes here in through the portal vein, and it goes into the liver. And once it's in the liver, then this portal vein breaks down into little capillary beds. And then it's going to go back and eventually come back, once it drains out of those capillary beds, into another big vessel and go into the vena cava. So this whole piece here, that the hepatic portal vein exists between 2 capillary beds. The 2 capillary beds that they're talking about is a capillary bed that's in the intestines. So you have these blood vessels where an artery brought blood into the intestines here. It got smaller and became a capillary bed. And in the intestines, that's where you would pick up your water and your nutrients and your vitamins from the food that you ate. And so that's happening at a capillary bed. Those capillaries get larger and become venules and the hepatic vein. And instead of this vein just leading directly eventually back to the heart, it then goes into the liver, gets small again, becomes another capillary bed. Then the vessels get larger and become larger venules, and then eventually those dump into the vena cava. So instead of a vein just leading directly to the heart, there I go. What is the matter with me? It's Friday. Sorry, you guys. You're probably now all yawning at home. I'm so sorry. It's not that I think this is boring. I think it's actually pretty interesting because this is, the only place that we see this type of pattern of blood circulation where we have a capillary bed in your intestines and a capillary bed in your liver, and what connects those two is a vein. Normally you would have artery, capillary, vein, and then back to the heart. We go artery, arterioles, capillary bed in the intestines, portal vein, capillary bed in the liver, and then back to the vena cava. And this is interesting because there are lots of drugs that when you take medications, and I'm talking about prescription drugs, they go into your intestines and they have to be processed by the liver in order for them to work. And so they're absorbed into the bloodstream at the intestinal level and then processed by the liver and then returned into circulation. There are other medications, however, if they go into your intestines, they'll be deactivated by being processed in the liver. And so there are certain things that you have to take in and ingest in other ways. Like we talked about nitroglycerin tablets, how they have to be put under the tongue. That's because the nitroglycerin, if it were swallowed, it would be deactivated in this process of being the blood being cleaned by the liver. Also, this whole piece here of the liver assists with blood homeostasis. Any excess glucose that remains in the bloodstream and goes to the liver, if it isn't being used, that excess glucose is going to be stored as glycogen, which is animal starch, by the liver. And so this is how your liver helps to maintain blood sugar and therefore homeostasis. Also, by cleaning your blood and detoxifying certain substances, you're maintaining homeostasis. So I strongly urge you to watch the Hasudigan video because it's interesting and it helps you to understand it a lot better. So that's hepatic portal circulation. Fetal circulation. So the structures for fetal circulation that I want you guys to make sure that you review are this, what they refer to the arterial duct. That's the arterial Ductus Arteriosus. Thank you. Ductus, Ductus, I think, Arteriosus. Forgive my spelling. The oval opening, which is the foraminovale. The venous duct, which is the ductus venosus. Ductusvenosus. Hopefully my fingers are right. And then the differences between the umbilical arteries and veins, and the placenta is the other exchange organ. So I'm going to talk about each one of these a little bit more detail. So please, fetal circulation wrap, you can watch that. I have another slide here. So blood arrives to the baby's body through the umbilical vein. And it is going to be, it's showing here a red line because this is going to be fully oxygenated blood. Where was that blood oxygenated? It got oxygenated by the placenta. So the placenta is going to be oxygenating blood, and it's going to, so we're going to come from the placenta and oxygenating blood. It's going to arrive to the baby's body through the umbilical vein. It's a vein because it's leading towards the fetus's heart. It's the term artery and vein is always going to be in reference to the baby, not the mother. So blood is then going to enter the baby's body through the umbilical vein, and it's going to head towards the liver. At the liver, you have two things that can happen to it. Some of that blood is going to go definitely through the liver and make its way into other blood vessels, and some of it's going to be shunted right away. Oxygenated blood is going to go away from the liver because it's not really functioning all the way. It hasn't come online yet because the mom's liver is doing all the work. And so this is going to be shunted away from the liver through the ductus venosus, and it's going to go straight up. So now here you have a mixing of the blue blood. This is the vena cava, okay, from the baby, and it is deoxygenated blood, and it's mixing with the oxygenated blood coming from the mom. So now instead of using just the traditional blue and red, this is why up here we've got the light purple, because this purple blood here, I changed to purple, is a mixture of the blood from the baby's body, which is deoxygenated, and from mom's body, which is fully oxygenated. In the heart, we have, remember, the right side of the heart would be sending blood off to the lungs, but the lungs aren't working. So we want to shunt as much of the blood away from the lungs. So we have two structures, the foramen ovale, which is going to connect the right atrium with the left atrium, so some of the blood can bypass right away. And then we have the Ductus Arteriosus, which is a branching between the pulmonary artery, which would have been going to the lungs, and the aorta. And so we have a mixing here. So notice again, it's still purple, so it's not fully oxygenated. When you're born, I'm going to clear off all these. The ductus arteriosus collapses, and it becomes something called the ligamentum arteriosum. And I'm sure I spelled that wrong. I think it's I-O. So the ligament, it collapses and becomes a ligament, okay? Collapses. Then the ductus of the foramen ovale becomes the, God, my brain dead. I've probably eaten really poorly today, and I'm feeling my sugar rush from this morning. It becomes, let's see, the foramen ovale becomes the, it'll come back to me. Foramen ovale becomes the fossa ovalis. There we go. Fossa, which means depression. Ovalis, O-V-A-L-I-S, which means oval depression. Okay, so you get a little depression. So we talked about if someone has a hole in their heart, it's not really a gaping hole on the outside of their ventricles. It means that they had the hole in between and it didn't collapse. And then the ductus venosus becomes, I think it's called the ligamentum venosum or something. It collapses and becomes a ligament, and it's not used. Okay, so those are those things. And then, of course, the umbilical arteries and veins dry up and fall off. You know, when you're born, that's what they cut when they cut the umbilical cord, and they have to put a clamp between the baby and the mom. They actually use 2 clamps, and they cut in between those 2 clamps. So they want to cut off the blood supply from mom into the umbilical cord, and then they also want to stop the blood supply from the baby out. If they just cut it and left it, wide open without tying it off between mom and the baby, then baby could lose blood, mom could lose blood, and that wouldn't be a good thing. So those are your circulation patterns, and this is just another picture showing all of those. It shows nicely where we have oxygenated, deoxygenated, and mixed blood. And then this is just another. So this is the end of page notes on 46, I believe, on circulation. Okay, our last page, our last right-hand page is page 48, blood pressure. And we talked a little bit about blood pressure when we did our lab, the Haspy stress lab, where blood pressure is the pressure of the blood inside of arteries every time your heart beats. And we measure that with 2 numbers, the systolic number over the diastolic number. And you probably, hopefully you remember taking those blood pressures. What's a good blood pressure to have? Well, 120 over 80, 110 over 70, 100 over 60. Those are all great blood pressures. It's the bottom number that we get most concerned about. When that bottom number, when you get blood pressures like 140 over 90, 160 over 100, it's the bottom number that we get so worried about. If you're exercising, your top number is going to go up during exercise, because obviously your heart's going to be beating faster and harder with more force, and so that's going to increase the pressure inside your vessels when your heart's beating. But it's the bottom number, okay? Your systolic over systole over diastole, your diastolic number. That is the pressure inside your vessels when your heart is relaxed. And so what kind of things would increase blood pressure when your heart should be relaxed? Well, the thickness of your blood. If you're dehydrated, your blood pressure can go up. Blood vessel resistance. What's going to resist? Well, if your arteries are hardened because of atherosclerosis or arteriosclerosis, any hardening of the arteries, any buildup of deposits, cholesterol deposits on the inside of your arteries, it's going to cause that lower number to be higher, and that's the one that we really worry about. So you want to have good, healthy vessels. Vessels need to stay elastic. They need to stay smooth on the inside. You don't want things depositing. So your blood pressure is going to change, but you don't want it going, that bottom number going up higher. Okay, and oh my goodness, is that the last slide? I guess it must be. Pulse. Pulse is the alternating surges of pressure. Okay, so we'll write about pulse. You're going to feel your pulse in arteries, and it's the surge of pressure when your heart beats. Typically, we take it at the wrist, okay? And we'll typically count pulses for, you count for like 15 seconds, and it's given as beats per minute, okay? Beats per minute. And a great pulse, 60 to 70 beats per minute. But if your heart's really racing, you can have a pulse of 120 beats per minute when you're exercising. The healthier you are, the better in shape you are, your pulse is going to come down much more quickly when your heart is in good condition. So, I'm sorry, I just wanted you guys to have a little bit more notes, and stay tuned for other things that I'll put up, other videos just to help you, because we've moved more quickly than I would typically like to, but we're up against that deadline of spring break. And so I just want you to feel that I've pointed you in the right direction to make sure you are as ready as possible. Okay, so I hope this helped a little bit. And anything else that I find that I think will help you to fill the gaps, I will put it up on Google Classroom for you. Okay, so I hope you are studying hard. Make those flashcards. Do your quick check questions. Do the chapter test and chapter review. Even if I don't assign stuff, those are all good ways to study, okay? This only took 17 minutes and 31 seconds, so I think that was time well spent for you. And I hope you're finishing up your notebook. Have a good rest of the weekend. Bye.